you so much for having me here. Can you see the slides all right?
Yes.
Okay, yeah, so today I want to talk about two different things. I will start talking about some work we've been doing on binding free energy calculations and comparing equilibrium and non-equilibrium approaches. And I will then give you a sneak peek on some work we've been doing more recently on an approach which we call separated topologies, which is aimed at broadening the scope of relative binding free energy calculations. And so free energy calculations are challenging, otherwise we wouldn't be here today. And there are multiple components that really impact the accuracy of these calculations. And I will talk about two of them today: slow protein conformational changes upon binding, which can be really large, like we saw right now in Julie and Michelle's talk, but could also just be a small sidechain rearrangement like here. And then I will also talk about the sampling of buried water molecules in the binding site. And so a really important question here is, how do we address these issues? And a lot of work is being done in this area, and it's really exciting to see how the accuracy already is improving. But there's another question that we're really interested in, and that's how can we detect these sampling problems and figure out what is going on in our specific systems in the first place. And I will talk a little bit about that today. And so binding free energies are typically obtained through thermodynamic cycles, and I won't go into the detail of this since we just heard that, but I just wanted to point out that in a couple of my slides, I will just talk about this part of the thermodynamic cycle where we turn off all the interactions of the ligand in the binding site, just because that's where we saw most of our sampling problems. Free energy differences between two physical end states can be calculated using different approaches. And a very common approach is the equilibrium approach. And there we have our two physical end states, and we define a set of intermediate states or lambda windows, and we perform a long MD simulation for each of them until we reach equilibrium, and then we sample the data at equilibrium. On the other hand, there's the non-equilibrium approach where we run the same long MD simulation in the physical end states. And we run through this chemical path in these fast non-equilibrium switching transitions where we switch the system from one physical end state to the other one in a single transition and calculate the work that it takes to go from one state to the other one. We do this a bunch of times and both in the forward and reverse direction, and obtain these probability distribution of work values, which are then directly related to the free energy difference through the Crookes fluctuation theorem. And what got us really interested into this non-equilibrium approach is that all of these fast switching transitions are independent of one another and thus can be run in parallel, which can potentially reduce the wall clock time of the simulation compared to the equilibrium approach. And so we wanted to compare these two approaches, and we started off with like the famous model system, the 2,4-lysozyme mutant L99A, which has this buried binding site that binds these apolar small molecules, like toluene in this case, which binds there in these two different binding modes. And so we run both the equilibrium approach and different protocols of the non-equilibrium approach that differ in the lengths of these switching transitions. And we run all of them for the same amount of total sampling time and look at the cumulative free energy difference here in the binding site as a function of sampling time. And what you can see here, that actually pretty quickly, all of those protocols converge to the same free energy difference with an uncertainty, which is really promising. And one lesson that we really learned here was that in order to compare the approaches and or also then further down to discover sampling problems, that it was really important to run independent repeats of the simulation. So here we're showing the mean and standard deviation of 3 repeats. And so that just helped us better assess the uncertainty, since the analytical error estimate usually underestimates the true error. We then looked at another ligand, 3-iodotoline, binding to the same p-lysine mutant. And from the literature, it's known that this ligand introduces this slow side chain rearrangement of this valine side chain in the binding pocket because it's a slightly larger ligand. And so again, we're comparing here the equilibrium and different protocols of the non-equilibrium approach. And you can see that in 400 nanoseconds total sampling time, those do not converge to the same free energy difference. And that especially this equilibrium approach has a high standard deviation. And although this is, of course, something we do not want to see, this really helped us here to identify some metrics that can help test for problems in free energy calculations. And so in the non-equilibrium approach, one thing we really like to look at are the work values for all of the transitions here, for the ones in the forward direction in blue and the ones in the reverse direction, orange, and their work distributions. And one thing that you can notice here, that here in the reverse direction, there's this jump in work values. And also the two distributions don't overlap very well, which indicates that there's dissipated work and some sampling problems. And so in order to see which slow motion in the protein in the end state simulation could be correlated with this jump in work value, we calculated the Pearson correlation coefficient between all of the side chain dihedral angles and the work values. And we found a high Pearson correlation coefficient between the work and this dihedral angle of the valine 111 side chain. And although from the literature, it was already known that this side chain is the one that really is a slow motion in the system, this helped us to identify this as a good metric, which then helped us to identify different sampling problems in other systems. Here we only see one transition of this dihedral angle, but also in the other systems, we then found multiple transitions, which then correlated well with the work values. Now, in the equilibrium approach, we have, like I said, all these different alchemical states, which we sample. And so we have a huge amount of data. And so it can be hard to see where and what our sampling problem is. And one thing that really helped us here was to look at the derivative of the Hamiltonian with respect to lambda. And this DHD lambda is computed with most simulation packages so that the user is then able to use that to get the free energy difference using thermodynamic integration. And so here I'm showing you for one of our intermediate lambda windows and the 3 trials that we ran. And you can see that sometimes there's a jump and a sudden change in the DHD lambda, which we then found that it correlated again with the dihedral angle of the side chain. And you can see that this rotation was not sampled appropriately in these trials. And so this really, in this case, led to the high standard deviation across trials. And this method can then also be automated, looking at and correlating slow motions in the system with sudden changes in DHD lambda, and really proved to be very helpful in multiple systems that we looked at so far. And then we wanted to move more towards more pharmaceutically relevant system. And so we picked HSP90, a known anti-cancer target. And the ligand that we looked at in the study binds to the solvent-exposed binding site and has, there are these 3 buried water molecules in the binding site. And here we ran the calculation slightly longer, total simulation time of around a microsecond. And it took quite a while for different approaches to converge to the same free energy difference. Again, here in the binding site, not the overall binding free energy. And standard deviations were also high, again, indicating some sort of sampling problem. And here we found that the sampling of the right amount of water molecules in the binding site was really challenging. In the equilibrium approach, we found that when the ligand is only partially interacting in these alchemical intermediate states, that more water molecules are able to enter this buried space, and that this motion is really slow. Here I'm showing you, again, for one of these intermediate lambda windows, the correlation of the DHT lambda with the number of water molecules in this buried space. And you can see that in this case, it took up to 40 nanoseconds for more water molecules to enter the space, while in others that happened earlier in the simulation. So that again led to a high standard deviation. In the non-equilibrium approach, we saw a little bit of a different picture. Here we saw that the to-work distributions really did not overlap at all, which shows that there was dissipation. And here we identified that when the ligand is non-interacting in the binding site, water molecules completely fill this space. And when we turn the interactions of the ligand quickly on in these non-equilibrium switching transitions, some of those water molecules get stuck in this same buried space, which then leads to the high dissipation. And so from the crystal structure, we knew that there should only be 3 water molecules once the ligand is completely interacting. But most of our transitions ended up with more water molecules in the space and only a few of them with the 3 water molecules. And so this really led to the slow convergence of the free energy estimate. And so far, I kind of gave you a deep dive into like common sampling problems in free energy calculations. And I introduced to you 2 different ways of how we can identify and test for problems. But overall, absolute binding free energy calculations are a very promising approach and has a lot of benefits. And the biggest benefit probably is that with these calculations, we're not at all restricted on the structure of the ligand, nor on the binding mode. So here you can see 2 diverse ligands. On the other hand, with the standard relative binding free energy calculation, we are highly limited by the fact that we can only compare the binding of related ligand that also have to have the same binding mode of a common core. And even some modifications to the protocol that allow for some scaffold hopping still mostly require that the ligands at least share a common core. And on the other hand, a huge challenge of the absolute approach is that we have to sample the unbound state of the protein and slow conformational changes upon ligand binding, while the relative approach that we do not need to sample the apostate of the protein. And so these things lead to the absolute approach being relatively expensive computationally, while the relative approach can be very efficient. And so this now leads us to the approach separated topologies, which we are developing in order to broaden the scope of these relative binding free energy calculations to allow the comparison of structurally diverse ligands. And so the separated topologies approach is also an approach of relative binding free energy calculations. And so, here we calculate the relative binding free energy by doing 2 absolute binding free energy calculations in opposite directions. Here I'm showing you the thermodynamic cycle of the binding site. Then similar has to be done in the solvent too. But here we start off with one of the ligands as a dummy ligand in the solvent, and the other one is interacting in the binding site. We then first insert this dummy ligand and restrain both ligands. We turn on the van der Waals on the green ligand, then charge the green ligand while at the same time turn off the electrostatics of the magenta ligand, then turn off the van der Waals of the magenta ligand so that it's now a dummy in the binding site, release the restraints and remove the dummy into the solvent, where then we perform the hydration free energy calculations. And so now why would we want to do that, right? Run 2 of those expensive absolute binding free energy calculations to the cost of only getting a relative binding free energy. And so one huge benefit of this approach is, since we are decoupling the entire ligands, that we are again not restricted at all on the structures of the ligand nor on the binding mode. But at the same time, since it's a relative approach, we do not need to sample the apostate of the protein. And so if the two ligands introduce the same slow protein conformational changes upon binding, those do not need to be sampled. And so from the efficiency, we hope or we think that it will be somewhere between the relative and the absolute approach. And so this method has been prototyped earlier in the lab. And now we're focusing on developing a software implementation that can then run these calculations on pharmaceutically relevant systems and on a large scale. And we use GROMACS as our MD engine and developed just a Python package to set up these calculations. And so we were able to run this approach on the first test system, and we chose tyrosine kinase 2 inhibitors from the Schrödinger JAX15 data set. And here you can see two of those ligands, and they share this common scaffold and only differ by small R groups. And so we chose this system to then be able to compare the separated topologies approach to the traditional relative approach, here calculated by Yun Hui in our lab. And so far we studied these two ligand pairs, and we were able to obtain the same delta delta G as the standard relative binding for energy calculations within uncertainty. And so one of the big challenges of this approach is that the choice of how we restrain the ligands in the binding site has a huge impact on the efficiency of this method. And so we've been investigating different restraining approaches, and I won't be able to go into the details of that today, but we will make our code available on GitHub. It's still undergoing major construction, but it will soon be there for you to try out. And so ultimately, we're interested in comparing the binding of structurally diverse ligands like these bromodiamine 4 inhibitors, which have been investigated with absolute binding for energy calculations. And so we're interested in these transformations that really fall outside the scope of traditional relative binding for energy calculations, and how now the separated topologies approach performs on these systems. And today I will have to end this talk on that cliffhanger, but I just wanted to say we're really interested in testing this approach on a variety of systems and really challenging transformations. And so I would like to ask if any of you, if you have some systems, some test systems that could really be helpful to us, we'd highly appreciate if you can share that with us, and that would help us a lot. Yeah, and with that, I would like to thank the Mobley Lab, and especially David, for really being a great advisor and for always supporting us. I would like to thank Bert and Vitas for all the help in the non-equilibrium approach, OpenEye for letting us use their software, and especially Chris Bailey and Gaetano for all their scientific advice, David Hahn for his work on the protein ligand benchmark and his help with providing input files, and funding from the NIH and Pfizer, and there especially Eric Dybeck and Vishnu Shrestha for their scientific advice on the separated topologies approach. And with that, I'm happy to take any questions you may have.
Thank you, Hannah, for such an excellent talk. There is the first question, so I'm going to read it out and copy it into the chat as well. So, hi, Hannah. Would you also consider starting the simulation with different conformations of the valine 111 sidechain other than the ones in the APO structure with respect to the ligand to test the convergence of the absolute binding free energies?
Yes, that's a very excellent suggestion. We actually started the calculations with the holo conformation of the valine sidechain. So we had to run long enough to look for it to reorient. But in this study, we did not check the convergence by starting it with the two different protein structures. But I know that other people have done that, and that's I think a really good way to check for convergence.
Great, thank you. So the next question is, hi Hannah, good talk. In protein ligand binding, some waters are important and some are less important, and this also varies based on the structure of the ligand. Is there any way to figure out how much energies a particular water is responsible for, absolute relative binding free energies?
Well, I think one way to test for that is to just sample just the water molecules, for example, using like methods like GCMC or the water map analysis to really check if waters are energetically, if they really want to be there, or if they are less energetically favorable. Yeah, so I think there are other methods to do that well. And you're right, definitely. Sometimes here in this case, we actually also ran these calculations, both having these crystallographic waters present in the starting structure and not having them present. And the absolute binding free energy really was greatly impacted by the fact if we had them present or not. So in this case, it really had a huge impact.
Great. So I've got the next question. Great talk, Hannah. Are you calculating the free energies using mbar, bar, or TI? And which would you recommend?
Here in this work, we used mbar to calculate the free energies, but we also actually, since you have the data there anyways, we also calculated it using TI and then used that sometimes to see if there was a huge difference between mbar and TI that also can give a hint that something is not right in the simulation, that there's some sampling problems. Typically, I think that mbar was shown in other work to be the most efficient estimator in general.
Then we have another question that has been upvoted. I'm not 100% sure how necessarily that relates to your talk, but in the star-shaped perturbation in relative binding free energies, which direction, forward or reverse, is harder to converge?
Yes, I'm not sure how to answer that. I would say that really depends on the ligand pair that we're looking at, that which direction would be harder to converge. But I myself have not run these calculations.
Yeah, I think I would second that answer. Here's the classic force field question. So, hi Hannah, great talk. What force field did you use for the calculations? Did you compare across force fields, and did you see any impact of that choice?
So in this separated topologies approach, like those results I showed you there, which shows the Ember99 SB star force field and I think open force field 100, and there we just chose this force field to be able to compare it to the results from the standard approach that Yun-Hui and all that calculated. But I do believe that these transformations were also calculated with the OPLS force field, but I have not yet looked into how our results compare to that.
Right, then just a very quick question from me. In terms of simulation time, how does it compare the separated topology versus the relative binding free energy in terms of effort?
Yes, so currently our separated topologies approach is still very expensive, so much more expensive than the relative approach, but we are looking into improving the efficiency, so both through the restraints, but also just reducing the amount of lambda windows. And so we want to run this approach on more systems to really be able to find a good general protocol, but that's also efficient. So that's something we're still trying to reduce the cost, but right now it's definitely way more expensive.
Great, yeah, that makes sense. Well, thank you, Hannah, so much for your talk. I'm going to do